Thanks, Aviv. That was pretty impressive. Very cool to add the spatial component. I didn't know this GeoMX, and it looks very high. The quality, it's good that you also go for other organs. I shouldn't talk though. I have now the honor of chairing the Q&A of the three speakers. Maybe please, you can just all unmute yourself and maybe show your faces. And we're going now to questions from Slido. And I'll start right away with the first upvoted question. So people just please go there if you don't have question, upvote or ask there. First question is from Malte Lücken from Helmholtz Munich for Andrea. Last week we heard about a different one autoimmunity being predictive of severe disease. Do you know, find some link in the variants to this?
Yeah, so that paper, I think we refer to the same paper for Jean-Laurent Casanova. They clearly look at rare variants using a more candidate gene approach. So we cannot really compare our result with them because we look at common variants. I have to say that we see a signal on interferon genes, the one on chromosome 21. So for the interferon gene, I think there seems to be some convergence. For the other gene that they look at, I'm not sure.
Cool. Thanks. Let's get started with basic. I mean, people were asking, what's ambient RNA?
Oh, so when you process a tissue, you know, things are not perfect. Some of the RNA gets out. And when you work in autopsy samples, which have waited for many, many hours sometimes, and as you can imagine, cell death is happening and the cells are bursting and the RNA gets out. That RNA sticks to things. And so as you process them through, and it really doesn't matter through which single cell modality you do this, but for example, if you use droplets, as we have in this case, some of the things that get into the droplet didn't get to it with the cell. They were just RNA that's floating around. And that leads to lack of specificity. So if you look at a cell type specific marker, like say CD45 or immunoglobulin A, and they should really be, you know, only in immune cells or only in B cells, and all of a sudden you find epithelial cells expressing them, you should be suspicious that it's actually ambient RNA. Now, cleaning it is very difficult, and CellBender is a particularly good computational approach to do that cleanup.
There's been other things like SUPX and so on with sort of forward mixtures. Is there a specific angle to it? I didn't know about the CellBender.
Yeah, and I should actually give credit to John Marioni as well. He's also an author on that paper. It's John Mertash and Stephen Fleming. Yes, it's a generative model that starts from an assumption, tries to model in the generative model every aspect of what we think contributes to issues in single-cell RNA-seq. And so it goes in this painstaking way through thinking about the PCR artifacts, the floating RNAs. Ambience is only one of the things it does. It also recognizes empty drops along the way.
Oh, very nice. So you really have a forward model for that. That makes sense.
Fantastically, I can say that since I had absolutely nothing to do with developing it, that it is a fantastically
and it's sufficiently fast
method, and it works extremely well.
Yeah. Very, very nice. Question to Roland and Aviv. How do the lung, from Richard Scheuermann, how do lung markers compare with blood markers? So do you see overlap? Could you maybe integrate something like that?
Yeah, I think there's quite some overlap. I think first place I think to mention is that the overlap of the most interesting immune subcell types are actually found both in the lung and also in the blood of those individuals, so like macrophage, particular subtypes of macrophages. And accordingly, the markers which I referred to, like CCR1, 2, and 3, and so forth, and its corresponding ligands, you find them also very prominently expressed in the blood of those individuals. Now, we are in the very lucky situation that we have, for almost all of our individuals, we have done both single-cell RNA sequencing in the blood and in the airways. We haven't done a systematic comparison yet, but since we have this comparable data, we'll do this systematically. But from just an observation, it seems to be quite some overlap.
So all I will add is, I think Roland said all the critical things. We have not dug into it deeply yet. Part of the reason we haven't is that we have also tried to really focus on cells that are very difficult to analyze otherwise, epithelial cells, endothelial cells, and so on. So it definitely was a little bit of a prioritization. But I will point out, and Chloe might be able, if she's still around, might be able to pop on the panel, that the Boston area team also has a major effort in the PBMC dimension And extremely sadly, since these are patients that actually were admitted to the ER, in our community and ended up passing away, they actually volunteered for research when they were still alive, and their blood was collected as well. We didn't know at the time, of course, but there's there might be one or a handful of such examples, and those would necessarily be anecdotal. One of the big challenges with these autopsy samples is that even with a heroic effort, you're basically getting to 20 patients and not much more. And so obviously the statistics are going to be limited. I think the BALS and the PBMC studies go to larger numbers.
I think in this case also the cross-tissue study I think will be fascinating. So
that is what we're hoping for. That is what we're hoping for. Kidney, liver, brain, lungs, livers, and more would allow us to answer some things about commonalities as well.
So just to add a few sentences about what Aviv referred to, we have collected samples from 306 COVID-positive patients over multiple time points. And so we do have patients that overlap between the autopsy cohort and the blood cohort. And this is all preliminary analysis. But not only do we see activated inflammatory monocytes, both in blood and the tissue, and as Aviv pointed out, we can also compare with heart and liver and kidney. Anecdotally, we also see some of these T cells that have homing chemokine receptors. And so we'll be able to compare the TCR profile and thus infer if it's actually really the same clones that are trafficking.
Yeah. And in addition, in the tissues, and Chloe is might want to comment on that as well, in the tissues there's also tissue resident cells, for example, alveolar macrophages and so on, which can provide additional immunological insight and tell us what is it that we cannot see in the blood and might actually still play a role. But I do think it's early days, and these data will be dug into multiple times until we have a better picture of what is going on.
Yeah, absolutely. We need a bit more time.
Fabian, may I comment on the ambient RNA, because I think it might be very different in the COVID-19 setting. So
please do. I was just about to say to all of you, I really like this discussion. It would be so much nicer to actually do this physically. There's a whole bunch of questions, so maybe if you could sort of.
Okay, I just shut up.
No, but no, you have to say this now. Just totally.
I think our ambient RNA is a carrying very useful information in the COVID-19 case, because obviously the infected SARS-CoV-2 infected cells, they die very quickly. And so we have actually turned around and looking into those cells which actually do not pass QC to actually have a better picture of the infection level of different cell types by SARS-CoV-2, just as mentioned this in passing by.
Going in the dangerous area of low QC cells, that's interesting.
Yeah.
Nice.
Yeah, absolutely.
I should just emphasize to Roland, one of the big challenges I highlighted is the long post-mortem interval, which is very different from the case of isolating tissue from living individuals. And so we simply can't tell. We can't tell if it's because they're dying from the virus or because they're more, they're the first cells to go when you just put a tissue and wait for 24 hours. And as a result of that, we just had to be conservative. We had no choice.
Going through some of the upvoted questions, there's a very nice anonymous one that states with Aviv, amazing work, and then focusing on the computation part, which I like a lot. This is the part, the computational session. So the question was if this transfer learning method that you used for automatic cell annotation, that's public, and in particular, if the manual cell annotation review that you then did, if it identified many errors of that.
Excellent question. So first, credit where credit is due. Graham Heimberg led the transfer learning automated work, and Racheli Normand, who's actually a joint postdoc with Chloe, has been doing a lot of the annotation you've seen for the immune cells together with Prisa Sen. There's also work like that being done in other cell subsets. The answer is kind of qualified. So Graham's method is not yet published because it was literally developed for this purpose, but a little bit by putting together several different concepts that are out there. And I didn't really spend time on that, but there were multiple our assumption is that there might always be multiple classifications out there that you also have to reconcile, multiple annotations that you're transferring, not just a single canonical one. So he's trying to grapple with that in the method. So similar to the meta-analysis that Fabian's team led for COVID-19, the necessity was COVID-19, but necessity is the mother of invention. You might end up with something that's useful for other purposes too. The manual annotations sometimes agree extremely well. I would say a lot of the time agree extremely well. and sometimes not. But these are also often in places, you might have noticed me being careful about saying fibroblast and myofibroblast, and we're not sure. These are places where also the automated annotation was not certain. And that's the nice thing about probabilistic methods, is that they don't just give you an annotation, they also tell you if they're confident in their choice or not. So in the places when there is fuzziness, you actually know, well, I should be a little more careful here. Maybe I should direct a lot of my effort of manual work in this particular corner where the automated method is unsure. And of course,
I think active learning or something like that will definitely be necessary.
Now would give us the opportunity of iterating again, because we've done the manual deep dive and the automated method, and we can start thinking about all the beautiful ways in which those could be combined. Thanks.
It would be nice for this CAP thing, for that platform.
Yes.
Also, if you have hierarchies or something like that, because you mentioned like fibroblast, myofibroblast, so those type of links would maybe more often be confused. Very nice. There was another question to Roland: if the shifts in cell types, if they're unique to COVID-19, if they're also seen in other causes of acute lung injury, and I think there was something about fibrosis, so on, that also was discussed already.
Yeah, I think, well, I have to admit, we didn't really systematically look into this, but anecdotally, I mean, if you look into the literature, I mean, many of these cell type shifts which we observed seem to be also observed in other acute lung injury kind of diseases. So what we try to do is now, with the flu season now coming in, to systematically compare also other viral infection of the lung to SARS-CoV-2 infection, to see what are the similarities and differences, for example, between H1N1, so the swine flu, and SARS-CoV-2 infections. And anecdotally, we have done this because there were some misdiagnosed patients which were thought to be SARS-CoV-2 positive, but they were found out not to be to have COVID-19, but H1N1 positive, and so we are now piling up these, additional patients to make this into a more systematic analysis. But we haven't done this really.
I think it could be wonderful sort of extension of HCA just being the healthy situation when you have those potential different diseases, maybe even automatically, well, or at least semi-automatically find some of those general covariate shifts. Very nice.
Yeah.
Let me throw out a question. Hi, awesome presentations. Aviv, Roland, even Catherine, in any of the viral positive cells, is there any evidence that the virus is controlling the cell, preventing it from dying? And is there a suppression of interferon signaling or NF-kappa B signaling in any of those cells that could suggest that the virus is really controlling the cell?
I'll answer from the autopsy angle. And again, Chloe might want to come online as well. I think one thing that I'm not certain about is whether what we're seeing is replicative. I mean, if you notice, most of the cells we see viral RNA in are not the cells you expect to be targeted by the virus for infection. They're not 82 cells. They're macrophages, for example. And that makes one wonder whether what you're seeing is clearing of virus versus classical, productive infection by virus. I would be hesitant as a result of that to interpret the gene expression patterns as a viral infection versus a viral response.
Maybe the organoids could be helpful.
Organoids could be extremely helpful for this. The BALs could be, there's many tissue studies also that could be very helpful for this. I think the autopsy setting is unusual in this respect, and it might be that the in situ data would also be more helpful for that question, but it's kind of an open question for us. And I know, Chloe, would you want to add to that?
Yeah, I think at this stage, it's really hard to, I think I agree with you. It's more a question of a response. I think we're seeing a lot of phagocytosis.
Yeah.
And not so much active infection. And some of it can also be related to technical biases because of the postmortal interval. We have necrotic tissue, which we excluded from the analysis. And the necrotic tissues were probably capturing the cells that were infected. So I think imaging could be also a better platform to capture.
That's why we're holding the nanostring analysis is so valuable to us, because it's done on FFPE and has some real advantages for this specific question. We'll also see what happens in the other tissues, where there we see RNA or virus at all, and endothelial cells, I think, are one of the biggest questions that we're very eager to understand better. And as you could see, there's different subsets of endothelial cells that are actually enriched with viral transcripts, and that might be the place where we will dig around that.
Pericytes too.
Other comments from Roland, Andrea on this?
So we looked into this very systematically, both on the epithelial cell side and also immune cell side. So for the epithelial cell side, we really systematically tried to disentangle the intrinsic from the extrinsic viral response. And this is actually a follow-up study of the one which I just showed. And you see remarkable differences in those cells, which actually seem to be directly seeing the virus in comparison to those which are indirectly, so to speak, seeing the virus because they are getting extrinsic signals through interferon signaling. So there's a bioarchive paper which you might want to look up where there's a lot of details about this. Now, for the immune cells, it's quite interesting. So there has been a lot of debate whether macrophages can be infected. And of course, we do find SARS-CoV-2 positive macrophages, but it would never, we wouldn't claim that they are infected, but they are just taking up the virus. And either by phagocytosis or by just, engulfing other cells. But interestingly, to your question, we do see that they are responding to the virus, even though they are not directly infected. So whenever they see the virus, they're doing something. And so it's quite interesting and fascinating, but it's, I think we are just at the very beginning of understanding what we are looking at.
I think my job is now to just thank you all again. This was a fantastic session. I really enjoyed the talks and discussions. It's so amazing how fast and sort of how integrative these communities have become, what type of studies are being done, like all kinds of things thrown onto these things. And Andrea showing collaboration with so many people that he can't fit it on the slides. This is truly impressive, and I really hope that there's interesting things coming up from that. Aviv and you are nicely agreed on actually following up potential additional questions on Slido, but now I hand over to Aviv and Sarah for closing the workshop.
Actually, John is closing the workshop. Jan Rendell, our Alliance, our Senior Alliance Manager for HCA.
Sorry for that.
Thank you very much. And I guess on behalf of everyone at HCA, I want to thank you all for attending today's sessions. This has really been a really wonderful session. And so I also want to thank our speakers and our moderators for some very inspirational talks. And I want to thank all of you for participating in the Q&A through Slido. I think we had some incredibly thoughtful questions and responses. And I know that the sessions both this week and last week have really spurred discussions about new collaborations, which is really, really terrific and really one of our goals for organizing these meetings. I also want to thank the program committee that helped put these sessions together, Sarah Teichman, Aviv Regev, Dion Taylor, Bruce Ernow, and John Marioni, as well as Brian and his colleagues at our AV partners, Allegiant Source, for their technical facilitation, as well as my colleagues in the HCA Executive Office, Tracy Andrew, Jenny Rude, Anya Hublaska, and Christine Schwenk. And finally, I want to highlight a few upcoming events, and we hope that you'll certainly tune into these as well. Later this month, we have our HCA Asia workshop that will take place over 2 days on October 22nd and 23rd. Then we have two upcoming webinars for our developmental biology seminar series, one on neurons in late October and early November, and then another in December on clinical and rare disease. And I want to highlight that we actually run these developmental seminars twice. So we have one edition timed for the Western Hemisphere and then another that is timed more for our colleagues in the Eastern Hemisphere. And the goal is really to facilitate participation from researchers all around the world there. And then after the first of the year, we'll be kicking off a new seminar series with our new biological networks. And so stay tuned, please, for more information on that seminar series as well. And information on all of these events is available on the Human Cell Atlas website at humancellatlas.org. Also on the HCA website, you can find information for joining the registries that you've heard about. Earlier today, you heard about the joint HCA Lifetime Registry for COVID-19 research, and that URL is up here on the screen, as well as the information for joining the HCA project registry itself. And we very much hope that all of you will consider joining both registries. Finally, if you wish to view today's talks again, or if you missed the talks last week, you can find all of these talks up on the Human Cell Atlas YouTube channel. And for our colleagues in China, they are also posted on our Bilibili channel as well. And we are always looking for suggestions for how to improve these programs, as well as ideas for future programs. And so we definitely want to hear your ideas. And so we encourage you as well to take our feedback survey. You can find this on the Slido website or also using the QR code that is up on your screen right now. So thank you very much. And with that, we will bring today's session to a close. Thank you again for attending.